If only the funding agencies would listen, right? It's very nice to be here. I thank Peter for inviting me out. So we've met at a couple of meetings in the past, and we're also manganophiles, but no manganese today. As Peter mentioned, I'm actually a chemist by training, and I've been doing chemical magic shows since I was a sophomore in college. And Sarah said, I wish you had sent me that one for the photo to put up. The other one was from the website. This is a photo of me getting a chance to drive the deep-sea submersible Alvin. And for those of you that may be interested, the Alvin is not in commission right now because we've just gotten a new ball fabricated. The engineering dives are supposed to occur in March, a scientific verification cruise in May, and then finally the new Alvin will be out for everybody to use if everything's working fine. And it's taken about 9 years to go from the planning to this stage. And it's not been an easy thing. I've been on the oversight committee, and the meetings on this have been amazing to the point where at one point they didn't buy enough titanium to build the ball correctly, to give you an idea of some of the things we had to go through. I'd like to start out with acknowledgments a little bit. The work I'm going to show you is due to several students in my lab, as well as Clara Chan's. Then postdocs, Greg Juschel, Sharon Kwan Crea, and Jeremy Rentz. And then my colleagues, Clara, Dave, Eric, and Bev Pearson. And then I have an industrial colleague. We've designed the microelectrodes that we use, and I'm going to show you a little something about, but he's always designed the electronics so he could deploy them in the field. So I just thought I'd give a little plug to him. And we've been blessed to have funding from a number of different agencies. A lot of the work here has been done through NASA, though, but some of it's been done with our Sea Grant program. So as an overview, this is going to be a story about oxygen and metals. And the metals are going to be iron and manganese. We're going to look at present-day sites as possible analogs of ancient oceans, if you will. Since I'm an oceanographer, I like to put it in that context. How fast do organisms respond to the absence or presence of these chemicals in the environment? And we're going to really focus on photosynthesis as one of the key issues here. How does oxygen affect metal chemistry? Again, manganese and iron. We'll talk a little bit about the thermodynamics and kinetics about these. Iron-oxidizing bacteria provide interesting iron-3 oxides that we believe are really neat biosignatures. You'll see sheaths and stalks in this talk from the organisms that Clara, Dave, and have been looking at. And we think that these are examples of ancient processes, and maybe we can look at some of these banded iron formations and make a guess, well, was it from cyanobacterial oxidation where the oxygen was being produced and the iron-2 titrated out? Or was it from the oxygen already present in the FeOB doing it? And I'm going to show you how we can determine in situ kinetics. Being a physical and organic chemist, I like to use as much of physical chemistry as I can. And since we now have tools that we can take in the field, it's nice to be able to do things in situ for the kinetics. So I'm going to do a little bit on our electrodes first. You see two designs here. The first one is glass. This is the one we're going to actually use for this particular talk. You'll see a gold wire going down the center of this glass, and then it's solder, carbon paste, or silver pasted onto a copper conductor wire, then onto whatever cable you need to go to your instrument. The top one is what we use for water column work, and I can take this and pound on it, and it won't break. So this is polyethyl ether ketone that many of you probably have used for HPLC techniques. And what you see in this is actually an epoxy. I'll show you now the tip of that, but what we're going to do is voltammetry, in particular cyclic voltammetry, and I'm going to relate this to how we do spectroscopy, so you see the analogy shortly. But we can measure every one of these particular species or determine their absence, which is just as critical. So if something's non-detectable at a particular detection limit, that's useful. In addition to this, we can even measure reasonable levels in the sediments, and I showed in Jim Bishop's class yesterday that we could actually measure lead in salt marsh soils that were contaminated at this about 5 micromolar level or so. So we could even measure zinc, cadmium, lead, and copper, potentially. The way the tip looks, this is one of our first ones. This is the 100 micron diameter gold wire. And you see the glass on the outside. And in the inside, we have a non-conductive epoxy from West Marine. And then what we do is we sand and polish this and it's almost optically flat. So we sand it with 4 different sandpapers from coarse to fine, and then 4 diamond polishes. Never use aluminum. If you really want good data, always use diamond. It's harder, gives you a better finish, and then it's better for the mercury film. Notice I just said mercury film. So we actually coat this with mercury. The total amount of mercury on this tip is 10 to the minus 10 moles, to give you an idea. If it were not for the mercury, we could not measure the metals, iron and manganese in particular, because they're found that they reduce at very negative potentials. We've actually tested this design to about 5,000 meters, 500 bar, and up to 120 degrees C. It turns out the epoxy starts to melt, or at least gets soft at 100, which makes it tough. We've tried peak inject. We've done some injection molding for the peak, and the peak doesn't start to get deformed until about 250. So we can actually go higher, but we haven't done that. So I wanted to show you some scans. Now, voltammetry, we're going to apply a potential between that working electrode I just showed you versus a reference electrode. And the reference electrode we use is a silver-silver chloride. We measure the current through a platinum wire. So this is a typical 3-electrode system. Chemists generally know about voltammetry. People that are not chemists don't, which is why I do this. And what you do is, if you look at wavelength versus absorbance, you're looking at absorbance on your Y, wavelength on your X. We're now going to look at current on the Y and voltage on the X. Or if you want, you can look at count versus energy, as you do with synchrotron radiation. In the long run here, what we're going to do is we're going to scan from a somewhat positive potential, minus 0.1 volt, negatively to minus 1.8, and then back. This is what oxygen in seawater looks like. It also looks like that in freshwater. This system works in freshwater as well. So we don't have any problems with salinity changes. It doesn't work in deionized water. You need at least a little bit of salt to make everything go. What you're going to see here is that this is a multi-analyte sensor, because you'll see here's sulfide. Sulfide's a film former on the mercury. So it forms mercury sulfide, and you destroy that film, and you get a nice sharp peak. So immediately you can see that the shapes are different, which immediately lets you know that you can discriminate them. But what's interesting about all these is that every time you see a vertical line, that's either the halfway potential or the peak potential. And that means that either an oxidation or reduction reaction has occurred, and it's specific for that analyte. So if you look at energy and counts, each element has its own energy, right? And so you can discriminate it. So that's how we discriminate the species. So here you can take a look at manganese in seawater. Here's iron in seawater. We now add sulfide and iron together, and you actually start getting another peak due to FeS nanoparticles in addition. So here's your iron, and here's your manganese. You notice that sometimes the shapes are a little different, aren't they? The manganese is sharper than the iron 2. And what you're doing is reducing manganese 2 to manganese 0 in the amalgam, and the iron 2 to iron 0 in the amalgam. Iron's harder than manganese, so it doesn't form as good a film as manganese. So what happens is you get a little different signal. If you take iron 2 and put it in slightly oxygenated water, notice there's no peak over here. You actually get a peak for iron 3. And these are iron 3 nanoparticles, usually stabilized by organics that are natural in the solution. Okay? So we don't have standards, unfortunately, for iron 3. But before I go any further, the detection limits are approximately 3 micromolar for oxygen, 5 for manganese, 10 for iron, and 0.1 for H2S in seawater. They're a little bit better in freshwater, which is what I'm going to actually focus on today. We don't have standards for the iron 3 or for the FeS, because they're nanoparticles. The other thing is that every iron 3 ligand complex has a different peak potential. So you can't actually quantify this, because if it's a different organic matter, the current versus concentration sensitivity curve varies. So that gives you a little bit of an idea. Now, people ask me about biofouling, because we put these out for a couple months at a time in the ocean. Because we're doing potential scans, and what I didn't tell you is that this experiment's done in 2 seconds. We scan at 2 volts per second. So we go 2 volts quickly this direction, stop in the sodium wave, and come back. So most organisms aren't going to try to get on the tip. In addition, it's mercury. All right? One other thing is you'll notice that we go off scale on a couple of these at the end at minus 1.8. We actually go into the sodium ion wave if we go in too far. and the sodium ion reduces the sodium amalgam, and then that generates hydrogen at the electrode, and that actually helps clean the surface. So we can actually clean these after every scan by scanning into the sodium wave. Same thing happens in fresh water. If you have potassium, calcium, magnesium, the same thing happens. So this is one of the ways that we clean the electrode. And there's other ways we can condition the electrode in between scans so that it comes back to its original condition. So these can go easily 20,000 scans, over a period of time. So let's talk about the processes that we want to study. Most of you know about banded iron formations. Here's an example that my student Robert pulled out of the literature. And the sources of the banded iron formations obviously are the silicon and iron to give the different colors. And they come either from landward, detrital sediments, hydrothermal sources. The question about oxygen can come from UV, can come from radiolysis, and of course it can come from photosynthesis. So for a while there, everybody said, oh, all the banded iron formations occurred because the cyanobacteria producing oxygen, the oxygen reacted with the iron-2, and the iron-3 precipitated. Now we talk about photoferrotrophs. We talk about iron-oxidizing bacteria. So now everybody's saying, well, which one was it? Well, it's probably all of them. The question is when and where, and what were the conditions when they were laid down. So Henrik Holland, of course, did a lot of this years ago, and he figured out that you've got quite a bit of iron, and he's got to hear his hematite, 66%. There's usually very low manganese in these deposits, and it's typically manganese 2, not 3 or 4, and it's usually low, 1%. These banded iron formations formed over about 1 billion or 2 years ago, or over a period of 1 billion years from 3 to 2 gigayears ago. And as I said, most people thought initially that this was due to cyanobacterial production of oxygen. So at one point, the gospel was we have low oxygen, high CO2 in the atmosphere. We have soluble metals in the slightly acidic ocean, and metal sulfides in the sediment. When photosynthesis kicked in with the cyanobacteria, where we have the carbon dioxide water going to sugar and O2, among other things, okay, what you now have is oxygen and ozone and CO2 in the atmosphere. It's going to take a long time for the oxygen being formed in the water by the cyanobacteria to get into the atmosphere. I don't think anybody's done that calculation yet, but I would argue with the amount of iron-2 and potentially sulfide it was in the water column of the oceans, 4,000 meters and deep on the average, that it's probably going to take a substantial period of time. It could be 1 billion years. Of course, we now have metal oxides and carbonates in the sediment, okay? And that's the way the present ocean seems to be for the most part. There are reduced systems, of course, in the sediments as you get deeper. So what's an analog of this? And we had the pleasure of working at Yellowstone National Park. How many of you have been to Choco Pots area? Anybody? Carl has. This is over by the Morris Geyser. That's iron-3 precipitates. And I'm going to show you how the system looks. And there's a fair amount of iron going into the stream from the source waters. So it's one of the few times that I've actually left equipment because we were almost attacked by these elk. And you can see that things are slightly in disarray. And what you see here is our lab-based equipment. This is the lab-based equipment. We were able to put it on 12-volt power, and we were using solar panels to regenerate the power of a car battery so that we could do all this. And we use a laptop much like this with a software to do all the programming of what we want to do and then the data analysis. So the way we have this is here's the source water. Here's a microbial mat. You'll see some green in here. And then you'll see what we call ferrihydrite here. And then what I'm going to show you now is some items about Choco Pots. First of all, the source is 55 degrees C typically. There's plenty of iron and manganese. There's no hydrogen sulfide detectable and no oxygen. So that means at 3 micromolar oxygen, we don't measure it, we can't detect it. At 0.1 micromolar sulfide, we don't detect it. So this is an iron and manganese-rich groundwater, but it's hot, or at least relatively hot. Since I study the vents, this is actually warm. So the way we set up the electrodes, have a micromanipulator. This is the electrode tip, and I get a glass blower to do it anymore. I'm not that good. I used to be, getting old. So I asked the glass blower to do this for me, and he can draw them down so you can have a tip that's fairly thin that far. That's great when you're going into sediments. In this case, it was a mat. But because we're going to do cyanobacteria, we could actually do dark light experiments. So we set up a tent. But we also could put filters in here, and I'll show you a filter that experiment we did where we only let 700 nanometer light through, which is where anoxygenic phototrophs actually start kicking in. So a blow up of this, you see the electrode, you see a PAR sensor and a manipulator, and then the reference and counter are just sticking in there underneath. You can't actually see them. Okay? Some people ask me, well, how far can you put away these electrodes? And I will tell you that in some of the work we've done at Vents, the working electrode will be where Peter is right now, and the reference and counter will be in the cold water over here. And we have no problems with an IR drop. So the way the mat looks is this way. This is some of Bev's work. And two organisms apparently are doing oxygenic photosynthesis, Synococcus and Cyanothes minervae. Chloroflexus, people think, has the possibility of doing anoxygenic photosynthesis, but it also does heterotrophic microbial geochemistry as well. And then ferrihydrite would be here. So you can see a little bit of everything in this particular slide. So iron-2 oxidation by oxygenic photosynthesis, we would take water as the reducing agent to fix CO2. You have two photosystem center, 1 and 2. Okay, and sort of a little cartoon for this, it would be this. Here's approximately the Calvin cycle, where the Calvin cycle is fixed, and photosystem 1 is generally where you take a little white lie here, but that's where you're producing the organic carbon from CO2, producing organic carbon from CO2 electrons and protons. Photosystem center 2 is over here. You need the light in a couple different places here with chlorophyll to take the water and take it to oxygen. This is photosystem center 1. Now, the phototrophs that are sulfide oxidizers and iron oxidizers don't have this. They don't produce oxygen. All right? They're anoxygenic. So Fritz Vittel and his group in 1993 published a paper about iron-2 with the photoferrotrophs, as they call them, and it only has system 1. So what we're going to do here is take away photosystem center 1, and basically what you're doing is that the iron-2 is pumping its electron out to somewhere else in the chain here, in the cascade, and then you get the Calvin cycle fixing the CO2. And so this is basically the difference, all right? So it's the light pushing the electron out of iron-2, you form iron-3, and that electron and other protons go to fix CO2. I should tell you that the organisms at the hydrothermal vents that we study use the exact same Calvin cycle to fix CO2, and those are not plants, okay? All right? So that's kind of interesting. At 3,000 meters down, snails, tube worms, mussels, clams, they're doing chemosynthesis with the same Calvin cycle. So I'd like you to keep that in mind. It's always kind of one of those interesting things about the evolution of life and processes. So what I want to do is show you some data from chocolate pods. So I'm going to show you a couple profiles. This one is one in the dark. Okay, and the red is iron. The green is manganese. Now you see error bars. With our electrode, because we can condition after every scan, we can run 3 to 5 scans and show you error bars. You cannot do that with any of the other electrodes that you commercially buy, like the oxygen Clark sensor, for example, because it consumes the oxygen so much that you just keep on getting it to go down, down, down. Notice that because it's dark, we don't have any oxygen produced. Okay? So what this means is that with no oxygen production, iron-2 and manganese-2 are not being oxidized. And notice that atmospheric oxygen is unimportant. The flux of oxygen across an air-water interface is really slow. All right, so we're not seeing any effect from that. Now, if we now go to a light system, all of a sudden you see that we're getting a peak at about 0.5 millimeters. This scales in millimeters on the y-axis. And we're right at the saturation point, and a little bit higher than saturation because it's 55-degree water. This is flowing water, by the way. So now we have oxygen titrated to the point where it's a maximum. It takes out all the iron. But notice you can measure iron above the air-water interface. All right? And there is some efflux of oxygen from the production of the oxygen by the cyanobacteria to the air-water interface, but it's not taking out all the iron. Notice that the manganese is still there. It's not being taken out. Now, in both cases, we measured the pH, and the pH is around 6. So we're going to explain to you why the iron-2 does get precipitated and the manganese doesn't. And this is one of the big things in the banded iron formations that I had at a previous slide. So basically, with the oxygen being produced by the cyanobacteria, okay, we oxidize the iron 2, but manganese, not manganese 2. So I want to show you what the whole scans look like for voltammetry. So here we are in the source water. Here we are at the MAD interface, 0.1, 0.6, 1.32. Here's where your oxygen signal is. We just reversed the way we scanned them in terms of plotting them. Here's your iron 2 and manganese 2, and people say, George, that looks kind of noisy. Yeah, it does when you have it like that. We do blow these up to get better measurements in the currents and the like. But remember, we're doing somewhere from 3 to 5 scans, and we're actually able to give you about 2 to 5% error bars, all right, which is pretty good, 10 at the worst, all right, and this is in flowing water. So what you see is a water-mat interface. The loss of iron 2 is you get the oxygen production. Notice that there's the manganese signal, but not the iron signal. Notice peroxide is almost on top of the iron 2, and we're starting to see a little bit of a bump for iron 3. We have to blow these up. This is just the full scan, just to give you an idea of what they look like. There's manganese is still present as you go deeper. And then, whoops, if I go back, you'll notice that once you get through that maximum part of the mat, now you're getting back to a manganese and an iron signal, and you're losing the oxygen. Okay? So that's how you saw that oxygen profile. Okay? So why does iron 2 oxidize and manganese 2 doesn't at pH of 6? By the way, Monday is the anniversary of Gibbs' birthday, of Gibbs free energy, J. Willard Gibbs. So what most people try to do is calculate the free energies. In this case, what we did is we took manganese 2 going to these different species of solids, iron 2 going to these, but we looked at only oxygen in the first electron transfer step going to superoxide, a reactive oxygen species. And what you'll notice is that this line right here, above this line is thermodynamically favorable, and below it's not. So this is just a PE-pH type calculation. And what you'll notice here is that at about a pH of 6, you still have the possibility of iron oxidation. But what about the manganese? Doesn't look it. Now, Peter may tell you that if you look at manganese 2 oxidation, you don't usually see that abiotically till about a pH of 9. And these calculations actually fall that way. All right? So again, this is the first electron transfer step. If you took oxygen going to water, both of these would be thermodynamically favorable. But this is what's controlling the oxidation, the first electron transfer step. And most chemical reactions will go by 1 electron transfer at a time or 2. Very rarely do you do 3. NO, nitric oxide, is one that could possibly do that. Now, if I now blow up a section here, what I'm going to do now is I have on this the oxidation rate in these circles right here versus the pH. So you notice that this is linear, just like the thermodynamics is linear. That means I have a linear free energy relationship between the kinetics and the thermodynamics. And as I tell my students, when you get one, cherish it, because what that means is you probably got something consistent with Marcus theory. And as it turns out, these are pi electrons coming out of the iron 2 going to oxygen. And that's the perfect one for Marcus theory. If you had manganese 2, it would be coming from a sigma orbital going to a pi and the oxygen. That is an unfavorable process, all right, because there's an orbital mismatch. So what you see here is the thermodynamics telling you why the iron 2 would be in abandoned iron formation. Now the question is, if you get a lot of manganese in abandoned iron formation, does that then suggest that the pH was high? And I don't know enough about all the abandoned iron formations and how much manganese is in them, but most of the time it's very, very low. So if you started to get lots of manganese, I would expect that that abandoned iron formation might have formed at a higher pH. So here's another way to represent it. This in case is now the Gibbs free energy versus the number of electrons transferred. So if I looked at the free energies of the free species, the hexaqual, iron, and oxygen, and manganese 2 and oxygen, I would have the sum of those free energies, and it would be here. If I now try to look at the first electron transfer step with oxygen going to superoxide and the iron 2 going to iron 3 or the manganese 2 going to manganese 3, you notice that manganese is unfavorable by a substantial amount. So that's what's going on. Notice, though, that every one of the other reactions that occurs, one at a time, with manganese 2 or iron 2, would be favorable. All right? To get one oxygen to go to one water, you need 4 iron 2s going to iron 3, or 4 manganese 2 going to manganese 3, if you will. And if you look at this little step right here for the iron, you would say, okay, this went up a little bit, but that's an energy of activation which could be easily overcome. All right? And that's just a simple way of looking at that. But this gives you a good reason why manganese 2 is not oxidizing. Now the question becomes, okay, we're getting this precipitation of iron from dark to light, all right, with the cyanobacteria. The question is, how fast is that? So here's an example where we put the electrode at 0.5 millimeters, right where the oxygen max is, all right? And now what we're going to do is we're going to make it dark, and then we're going to make it light. Within 60 seconds, at a normal day, 620 watts per meter squared, today would be in that category, you could take out all the iron. And this is 65 micromolar iron, too. All right? So what you see is the iron oxidation. You let it sit for a while, and then you go dark. And then, by the way, the iron oxidation rate here is totally inorganic, because if you look at it from the temperature, it had to be extrapolated a little bit because people haven't really done that at 55. But this is totally abiotic. That means that the oxygen being produced by the cyanobacteria abiotically or inorganically reacted with the iron 2 and took it out. All right? Because there's no use of this by the cyanobacteria, no use of the iron by the cyanobacteria. Now, if you go dark again, it takes a little longer for it to go back. The iron 2 returns to the mat. So this is a dark, light cycle. This is what happened. All day you'd be taking out the iron, and at night it wouldn't be taken out. It would be going down into the stream. So the question is, everybody likes to do kinetics. Well, what's the first thing you do? Let's do a pseudo first order plot. And that doesn't look like a pseudo first order plot to me. So if you just look at that decay, it's not a first order plot. But if you plot it as a 0th order plot, it's perfectly linear. Okay? So most of the time you say, hey, things got to be sort of like pseudo first order. Well, it turns out a 0th order reaction is consistent with light being the primary species of interest. Because if you don't have the light, you don't produce the oxygen. So when you have a 0th order reaction, something else other than the concentration of the species is important. So it's got to be the light. All right? So can we verify that? Well, yes. So we're going to do experiment. There was another time we went out, about 120 micromolar iron. And we're going to increase the light intensity with our 10. All right? And what you're going to see here is I increase the light intensity. The iron 2 goes down. And at 54 watts per meter squared, the iron starts to oxidize. And then here it is. At about 85 watts per meter squared, which would be a typical overcast day. When I was picked up the other day by Jim Bishop, it was a pretty overcast day. You would still be able to take out all the iron. Now what happens is you increase the light intensity upwards to what we would now have today. You'll notice you get this kind of curve. And it's a typical exponential growth curve that you would see for something that's light dependent. And it's a pretty good fit. And so this tells us that this is a light dependent reaction and everything's going well. At some point you're going to shut down photosynthesis because they're going to be inhibited. Now remember, this is the light that's actually right at the surface of the mat. It's not 0.5 millimeters below. It's right at the surface. But this gives you an idea of what's going on. Okay, so the question is, do we have any anoxygenic photoferrotrophs? Well, what we know of the photoferrotrophs, they will use the near-infrared around 700 nanometers. The cyanobacteria don't use that light. So here's a dark experiment in blue and a near-IR experiment in red, and they overlap. So the indication here is that the chloroflexes, although they're there, do not apparently do any iron 2 oxidation that's anoxygenic. All right? So that means that everything here in this case is due only to the cyanobacteria, not to anoxygenic ferrophototrophs. So I'm an oceanographer. We decided to do this one. You'll love this. We did this in the parking lot. So we took the mat, and what we did was we threw it into a little round bottom cylinder. We made an electrode which had all 3 electrodes in here. We had a temperature sensor here with a water bath. We injected the iron 2. We had an argon purge as we needed. We had a light sensor. And so we were going to mimic it like this was an ocean. Can we mimic it like it's an ocean? All right? A little mini ocean, if you will. And so we're going to show you some similar zero-order kinetics, regardless of how we do the experiment. So in this case, the light intensity varied. We were in the parking lot. We had some clouds come by, and you can see some variability in the light here. We started out with about 150 or so micromolar of iron 2 and about 50 micromolar of manganese. You notice the manganese through this whole experiment of 15 minutes never left. It stayed in solution. And what you see is these linear regions right here. And these linear regions, whether you do zero order or pseudo first order fits, they're the same. Well, if they're the same, then it's a zeroth order plot, not a first order plot. So this again shows us that no matter what we do, when we're doing this in natural light, this is not lab light, this is natural light. And you can see how fast these oxidations are going. Okay. Big key, again, the manganese is there. We kept the pH at 6. It was buffered slightly. We actually used some of the water from Yellowstone National Park that we brought back with us. So let me give you a little overview of this. For photosynthesis, we're producing oxygen. It's taking out all the iron 2, but not the manganese. And even a cloudy day is enough to take out all the iron 2 in these waters at about 150 micromolar. That's the highest we actually had. The iron 2 loss is zeroth order. And this indicates that we have an abiotic reaction caused by the oxygen derived from photosynthesis. And the iron-oxidizing organisms would yield first order, as I'm going to show you shortly. Okay? Also, they work both in the dark and the light. All right? Manganese 2 is not oxidized and removed at these pHs. Agrees with the thermodynamics. So we kind of feel that this system was really nice to give us an idea about possible models for Archean or Precambrian band iron formations where oxygenic photosynthesis would have been the primary process titrating out the iron. And of course, these filter experiments in the IR showed that there was no anoxygenic photosynthesis occurring in removing the iron 2. Okay? So now what we're going to do is we're going to switch to iron-oxidizing bacteria because that's a really hotbed of work right now by a lot of people. This is not far from our main campus. You can see these areas where groundwater comes out. Sometimes you can see some orange and yellow colors. This you might be able to see a little bit better. There's one of our microelectrodes in. And this is actually what our scans look like on the monitor rather than replotted. So this is oxygen with some manganese in it. The iron's already been titrated out because it's in an area where we're not at low oxygen. Now what we're going to do here, okay, is we're going to talk a little bit about biomineralization first. FeOB, not all of them, but most of them, give stalks or sheaths. So we're going to show you these unique morphologies. People like Joel Banfield and Clara Chan and others are talking about these being biosignatures for modern and ancient FeOB. And what we're going to do is show you, here's stalks from a seawater version of a FeOB. Here's a stalk from a freshwater version, somewhat similar, despite the fact one works at 35 parts per thousand salinity and the other basically at 0.1. You see the same types of morphology. Here's what a sheath looks like. It's actually got a rod. The organism is actually in the sheath and comes out of it, which is kind of neat. So these are the kinds of biosignatures that we're looking at right now. Talk a little bit about how they form this as we go on with this presentation. You'll notice little precipitates right here and there. That's the amorphous abiotic precipitation, because even though oxygen's present and the organisms are present, there's still chemical oxidation going on. So we want to discriminate between the chemical and the biological, which is most prevalent. As you'll see, it depends on the conditions that you provide. So you notice that there is still some amorphous precipitation going on, and that we believe is entirely due to chemical oxidation, whether it's chemicals in solution or chemical with autocatalysis with iron oxides. All right? Are we okay? Any questions? All right. So how do we study FeOB? What people started doing, Dave Emerson was one of them that started this with Eric Roden. They started doing gel gradient tubes with microelectrodes, mainly oxygen, to study oxygen gradients. But then when we came along, we could now do the oxygen and iron opposing gradients. And then we can do pure cultures. We have electrochemical cells. We can put the cell in culture. And we can actually manipulate the oxygen concentration to some low value. It could be like 10 micromolar, for example, to higher values. We can actually go lower than that. We've actually done some work. We've taken field samples and thrown them in a little vials and done the experiments. So I'll show you these experiments. And then we're going to show you the latest stuff we're doing, which we're ready to submit. Microslide growth chambers where we couple optical microscopy with the electrodes so you can actually see the growth of the iron oxide, see the directionality of the iron oxide to the opposing gradient. All right, and that's how we're going to end this section and wrap things up. So here's what a gel would look like. Typically you use FeS as your iron source, and that's usually at the bottom. And then you have your medium in here. This is an iron-3 growth band in this case. You have oxygen above, iron below. In this case, you'll see the oxygen basically going to 0 at about 2 millimeters. The iron is overlapping. It goes pretty much to 0 at the surface. All right? And right here is where the bacteria are, and that's where the band is. All right? So if you don't put it in an inoculum, you don't get a growth band. You just get indiscriminate precipitation. And that is amorphous versus the different kinds of morphologies that we do get. And so in this case, you don't really get a growth band. You don't have the bacteria concentrated in one spot. You just see oxygen and iron overlapping. Nothing special. All right? Pretty straightforward. Now, Greg Druschel, when he was in my lab, we tried to do things where he actually made about, in this case, about 10 micromolar oxygen right here. We're now going to do an experiment where we add the iron to, measure the oxygen and the iron. The red is an iron in azide, and the black is an iron live. So we do an experiment live, then we put in azide, do the experiment killed, and you can see a difference. Now, this doesn't have as much curvature as you normally would see in a pseudo first-order plot, but it's there. So we have the abiotic decay rate in red, because there's no biotic, and the black is the mixture of biotic and abiotic. And so we subtract the two to get these measurements. So you have to have exactly the right amount of cell counts to do this experiment. You can't have variable cell counts. So the abiotic rate equation has been known for years by a number of people. This is it. So this is without any pseudo, any kind of autocatalysis with iron oxide surfaces. So you have the rate constant K, and which varies with T, times the concentration of iron 2, times the concentration of oxygen, times the hydroxide ion concentration squared. That can be calculated regardless, as long as you know the pH. So you always know that number, what that number should be. All right? Now, if I do pseudo first-order plots, what you'll see here for the red, which is the azide killed, you notice that the rate constant for the decay is about 60% of what the rate constant is for the abiotic and biotic. So you can see that this is much faster when the organisms are present right there. Okay, so we can now discriminate those. And we can now say what's the percentage of oxidation due to abiotic and what's the percentage of oxidation due to biotic. So this is what Greg came up with. So here's his percent biotic rate versus oxygen concentration. I wish he had done a couple more. We're going to fill in some things with different organisms. But the take-home message for this organism was that at about below 50 micromolar, we could definitely see that there was a decent biotic rate. Once you got above that, with the one data point, it was tougher. We've done a couple other experiments to show that, but we're going to try to fill it out with about 2 or 3 different organisms, because we have found some organisms still doing well at 80 micromolar. Okay? One being Leptotrix, which Dave Emerson's been working with. So this gives you an idea that the organisms are able to compete with the abiotic rate once the oxygen level gets lower. So remember the chemical oxidation equation, right? It's the concentration of iron times the concentration of oxygen. As the oxygen concentration goes down, even if the iron initial is high, what's going to happen? The rate will go down, right? So we did some other experiments with other organisms, and this was with Dave Emerson at places over in Virginia before he went back to Maine. This is Gallionella. This forms nice stalks. And then here's a Leptotrix, which is forming the sheath. And what we did was we did an experiment at these 2 sites in October and December. And Jeremy Rentz figured out what's the percentage of oxide morphology from sheaths, what's the percentage from the stalks, and what's the percentage from amorphous. So you could actually get an idea of the relative oxidation rate from that characteristic. Now, that was all done by microscopy, but gives you an idea. And you'll notice there's a variability. Notice that the sheaths were very low in this case for Dragon Ridge. Notice that in this case, they went down, but they were still measurable. Notice that the chemical amorphous is relatively the same between these sites. pH was about the same. So it's a matter of what's the concentration or a number of cells per milliliter that's going on, because we just took the mat. So in this case, 65 to 75% of the iron 2 was biotic. When you started adding all this up, just from that kind of data. That's even better than what I showed you in the monoclonal culture. So this is actually in the field. So what Jeremy did was we actually redesigned a little vial. Always try to do things so you can do things out in the field. And in this case, we try to keep the oxygen about 20 micromolar. That's the green here. We have an experiment. We started out with iron live. So we put the mat right in, and you can see this decay curve. Beautiful pseudo first order look. We stopped the experiment, said, okay, add the azide, and we still got oxidation. All right? That should be only due to abiotic or inorganic chemical oxidation. So here's your rates. You notice, again, we've got something in the order of this 0.051 to 0.0883. So notice that we have a pretty good rate here. Again, it's not zero-order kinetics. This is really nice. You can see the difference between the 0th order and the 1st order. So the 0th order in the cyanobacteria doesn't show up here. Again, this is a fairly substantial amount of biotic oxidation compared to abiotic oxidation. So what Jeremy did, he took a lot of our data, and what he did, he said, okay, I can now calculate the oxidation rate for cellular activities in black, iron oxide catalyzed, that's the azide killed. But if I use this equation, I can calculate what the actual chemical oxidation would be for the aqueous reaction. And so what you see here, we know the pH, we know the iron content, we know the oxygen content, so we can do that very straightforwardly. So the chemical oxidation is the green, and you'll notice that it's fairly low. There's only one instance where it actually reaches something that's pretty significant right here. All right? Again, to date, all FeOB tested show first order kinetics. And in this case, you can see that, again, we're at the 75 to 80% oxidation rate for most of these. So what I'd like to do is shift to the last section that I said I would do. I want to try to get done so we can have time for questions. Here's Clara's optical microscope. We're going to put on our electrode in here with a slide. And we're using the seawater strain that we have. Everything else that I showed you to date was freshwater. So we're going to switch gears and show you a seawater strain. And this is what it looks like. This is a 50-micron gold wire going down here, sanded and polished and plated, as we said before. And then there's your optics. In this case, what we're going to do is the following. Here's a nice little cartoon. Here's your FES in here. You get two slides, one on top of the other. There's maybe 2 millimeter distance separating them. He puts Vaseline and other things around the side so oxygen won't penetrate. We put the reference electrode and the counter electrode on the sides, and the working electrode with a gold amalgam down the center. In this case, what we found was best was actually let the stage move so that the working electrode was stationary and the reference and the counter would actually move. And so actually we found that it was better control using the stage of the microscope rather than using a micromanipulator. So the first time we put in too much inoculum. And we're going to show you what happens when you do. This is about 10 to the 9 cells per milliliter. Here's your meniscus. So that would basically be about here. Here's your FES up top and then down below. You can see a band right here. That's where the band, and that's where most of the inoculum was. The electrode path went this direction. This is a 2 millimeter marker here. And then we made measurements about every 1 millimeter until we really got into the gradient. And as you can see, we did 2 different experiments with a microelectrode at 2 different spots, and you can see that we get very good reproducibility between the two. Okay? So it says to us, at least we're setting up gradients much like we would do with gel tubes or other types of systems in general. Now, it turns out because the inoculum was so much, we produced so much iron oxide, we got a really thick growth band. And as the electrode went through, it pushed the iron away. Okay, the iron oxides away. So just for kicks, let's show you that. So here's the tip of the electrode approaching. Here it is reaching the band. And here it is penetrating right through the band. Okay? And all the way through. So this is what happens when you have too much inoculum. So this has been taking a long time to figure out exactly what's the best so that we don't have some of these disruptions, but it's giving us an idea. But even with that in mind, if we set up a gel tube with the same type of cell counts, this is the flux of oxygen without organisms present, or for a control tube. Here's the gel tube set up with a, so this one had no, pardon me, the white has no organisms. The gel tube here, here's your oxygen flux. And then the two oxygen fluxes calculated from those two profiles really agree very well with the gel tube. So we know that what we're doing with these slides is similar to what we do in a gel tube. Okay, so that was nice because that said we're on the right track. So here's what a profile looks like when you're using like 10 to the 7 cells per milliliter. So when you first set it up, it's going to take a while for everything to get to what you would call a steady state. And what happens is you see the oxygen content go down, and here's your iron going down, opposing gradients. All right? What happens if you do it after about 8 hours? Okay, now you see your oxygen going down very nicely in a gradient, much like you'd expect in a sediment or mat profile. Here's your iron. Notice a boom, it crashed really quickly. Well, why did it crash? Well, it turns out that blue line is about 28 to 30 micromolar of oxygen, and that's where we first see the stalks forming. Okay, because this organism forms stalks of iron oxides. That area right there, which has all these X's, that's where we have the most stalks. And it turns out that the oxygen content in that area is from 3 to 27 micromolar oxygen. So these are really efficient forming stalks. You'll see a little bit of abiotic precipitate, but most of this is stalks. So we actually are showing that at this point, we're at about 3 micromolar, and at that point, we're about 27 micromolar. That's pretty interesting. So we were ready to submit this now. So we're actually fairly pleased with this. I hope you are. If you're not, let me know so we don't submit it and have bad reviews. But here's an example of some interesting things about the morphology, which Sean has spent a lot of time doing. So here's your band. So he's going to look at how these little pieces of precipitate line up. So what he's done is this. Here's a section here of about 10 micrometers. And you'll notice that there's an awful lot of directionality. They sort of look like they're going horizontal, don't they? So we see distinctive features. We'll see some branching. Here's some branching. You'll see twisting. And the directional growth is towards the oxygen gradient. All right? So they're probably manipulating their environment with the iron and the oxygen. Okay? But we don't know exactly how they're doing that. So there's the branching. And then he calculates the directionality. So sometimes you might see it go a little bit this way or a little bit that way, but principally it goes right that way where the oxygen gradient's going. Okay, and he has a special program that people have been using for this. So most of the direction is towards that oxygen, which is kind of interesting. Then he took some microfossil filaments and did the light micrograph. This is from a hydrothermal iron silicon deposit that's about 170 million years old. And the microfossils here have similar shapes as the stalks that we just showed you. They have some branching. Whoop, I thought I had an arrow there. No? You can see some branching. Yeah, there's one right over here. Right there. You can start seeing branching. If you really look at these clearly, you see branching and twisting, and you'll see the directionality seems to go in one direction. Now, we don't know what the gradient was, but you can see that they're definitely going towards one direction more than another. So this is a 170 million year old deposit showing similar types of characteristics as something that's brand new that we're seeing under the microscope. So we think we have a handle on this now. And these biosignatures, in this case, would indicate that this was probably a microbial iron deposit rather than a chemical iron deposit. And certainly cyanobacteria weren't doing this because this was the bottom of the ocean. All right? Or probably not. Okay, so I get two slides for conclusions. One that's in words and another that's sort of in a cartoon. We believe we can distinguish 5 different iron-2 oxidation processes using real-time measurements. I'm not talking about just the microscopy, but the real-time measurements. First of all, we know about abiotic or chemical without iron oxide catalysis because we know that equation. We can always calculate that. If we add azide, we can distinguish abiotic with both iron 3 and with autocatalysis from iron oxides and the chemical. We can do biotic and mediated by FeOB. Here, different FeOB will yield different iron 3 hydroxides, but in this case, we're going to see a pseudo first order kinetic. The fourth one is abiotic and mediated by cyanobacteria in the presence of light. And remember, all these 3 can go both in the dark or the light. Processes 1, 2, 3 follow a pseudo first order kinetics. Process 4 follows 0 order kinetics. Anaerobic phototrophs, also known as photoferrotrophs, that oxidize iron 2 with light, they don't produce O2. Cyanobacteria will produce O2 in light. These won't. Now, we haven't actually done that experiment yet, but other people have done some of those experiments. We like to confirm it with our electrodes. So far, the only biotic process mediated by FeOB yields exquisite morphologies. None of the others seem to give these exquisite morphologies. But not every FeOB will do this, but most of them that we've studied have done that. So the cartoon I have for discrimination of the pathways are the following. We have those reactions that occur in the light. Cyanobacteria versus photoferrotrophs. Well, we can distinguish between them, as I'll show you in a second. And then there's those that occur both in the dark and the light. There's the FeOB on the left versus the chemical, whether it's with or without iron 3 catalysis as the oxides. So if we look at this, cyanobacteria produces oxygen, 0th order iron loss, no biosignatures, not near IR active. No oxygen production for the photoferrotrophs, 0 order loss, no biosignatures from what we can tell, near IR active. Very easily discriminable to discriminate. FeOB, no oxygen production, 1st order iron loss, get biosignatures. Over here you get no oxygen production, 1st order iron loss, no biosignatures. So you can actually go out, and I've said this, I'm getting too old, I can't do this. So if is there a student interested in iron oxidation? You interested in a NASA project where you can put these electrodes on a lander where you might have water on another planet? You can now design a set of experiments with the proper light, right, dark, near IR active, and you could actually go on a mission, put these electrodes into the water, and if there was iron, and you could actually then either pump oxygen in, all right, or not, depends on what you want to do. You can then discriminate whether you have organisms there that are doing biotic iron oxidation, and you could actually prove that there would be life from the iron side. So I throw that out. I'm too old to do that because these NASA missions go, 20 years to the future. I am now too old to do that. I throw that out for somebody to do. Okay? And with that, I'll entertain any questions you might have.
It's interesting, as you pointed out, that these aerobic iron oxidizers have an optimum at a very low oxygen potential, 15, 20 micromolar. And has anybody looked into, and it's not an accident, I guess that's where they compete the best with abiotic processes. But has anybody looked into the physiology of how they actually optimize their activity at such a low potential?
Not yet, as far as I know. I know that that's one of the things Clara would like to do at some point. She and Dave and Eric have been talking about that. They're not generally on the physiology side so much. So they've been bringing in different postdocs and like into the group to start to try to look at that. So it's one of our big wish lists. So that's a great question.
Hi, that was a great talk. I I was interested in the initial part of your talk where you were discussing about the selective removal of the iron relative to the manganese in a mixed system. What about the interactions between iron and manganese themselves, excluding oxygen?
Yeah, so people have asked questions like that. How about nitrate? Probably there's not as much nitrate, but it's small enough that it's in the level of oxygen that nitrate could be an oxidizer for iron too. We haven't done that experiment yet in the lab, but people have done it generally in a test tube, if you will. The other thing is I don't know if the organisms will use the nitrate to do that. Iron 2 does react with manganese dioxide relatively quickly. Dika Postman published this back about 20 plus years ago, and he saw iron 3 precipitates and manganese 2. So the question then is, which is first, the iron oxidation or the manganese? Well, it turns out manganese, to have oxidation of MnO2, you would have to have a high pH if it were chemical, or you have to have microorganisms. Brad Thibault and Ken Nielsen have done work on this for a long time. And that's definitely a microbial oxidation when the pH is below 9. So they've shown it with spore coats as well as the organisms themselves. So my guess is on a geologic time frame, I wouldn't anticipate MnO2 being important. But on a modern time frame, where you do have seasonal activity in different systems where manganese oxides might form, and then iron 2 gets into that area, then that would be a possible oxidation. Now for us, that would be easy. For example, if there was no oxygen, what would happen is we would see the iron go away and the manganese 2 increase. So we can measure that. I don't have a probe for nitrate, so I couldn't tell that one. But I could tell that in terms of a...
But I guess that is what you saw. You saw the iron going away and the manganese not.
Yeah, but I didn't see the manganese grow. So if I had manganese dioxide and the iron 2 reacted, then the manganese 2 would increase.
I thought the manganese was always in the Mn2 stage.
Pardon?
I thought the manganese in your system was always in the
it's all
Mn2
manganese
stage.
Too, but I'm saying if you
so
had...
it was transient, so you wouldn't have seen it.
No. But if I had MnO2 present, okay, and there was no oxygen available, and then I would see the iron 2 go down and the manganese 2 increase, I could then indicate that manganese dioxide might have been the oxidant for iron in that case. But we didn't see that kind of condition.
Okay.
Okay, that's a great question. It's one of these really interesting things because we can measure both of them. If we're seeing the one decrease and the other increase, then it's like an opposing gradient with the oxygen and the iron. It's similar. But in that case, the manganese was always constant when we did the cyanobacterial experiments. Yeah.
In terms of your thermodynamic analysis comparing iron and manganese, what's the basis for only looking at the first electron transfer? Is it that the second one is too slow to have an impact thermodynamically? Or?
No, superoxide, and iron should go. The thing is that usually if you have a problem with a reaction, if in this case it's a thermodynamic control because the manganese 2 won't oxidize. As a matter of fact, let me do one thing. Those of you that know me, I like to do this. This is the interaction of iron with oxygen. Okay, this is a pi electron, T2G, if you know your crystal field theory. And that's a pi star electron. And so what you're doing is you're pumping an electron from iron 2 to oxygen. And this is thermodynamically favorable, but it's also kinetically favorable. In this case, for manganese, you're going to take an electron from a sigma. So you actually have a different orientation. The pi electrons are above and below the plane or the bond axis. So if I have a sigma electron coming in on the bond axis, it's not going to interact with the orbital, so I can't have electron transfer. Outer sphere. So for manganese, the process would have to be inner sphere, and when you raise the pH, you get manganese hydroxide rather than manganese 2-hexaaquo, and then this becomes favorable. So now it's an inner sphere process. So there's sort of this kinetic thing. The first electron transfer can be the controlling feature. In this case, it's a thermodynamic control if it's an outer sphere process. And that's what it looks like at low pH. As the pH increases, it's no longer iron 2-hexaaquo or manganese 2-hexaaquo. It becomes a dihydroxy manganese 2. And then you have an inner sphere process where the oxygen actually binds directly to the manganese 2. And then there's electron transfer. And it's only to that point where you get that. And that's why the thermodynamics starts to kick in at 9, because that's when you get the dihydroxide species. So you're looking at the speciation of the manganese in that case as a key. Does that answer your question, Carl?
I think so.
Right. We didn't try to do that. We could measure peroxide, for example. The way we've done that in the past, and it usually works when it's lower concentration than when it's at the maximum. The iron 2, the oxygen reduces in a mercury electrode in 1 2-electron step to peroxide. Then that peroxide, which was formed by the electrode, then reduces to water. So you see a ratio between the peroxide signal and the oxygen. If, for example, the peroxide signal increased dramatically over what you expect it to be, you could actually say we have peroxide. We've actually seen that in salt marsh sediments at low oxygen content, all right? And we've even seen it in a microbial map with sulfide oxidizers, okay? And cyanobacteria layered on top, one on top of the other. But it's usually difficult for us to see it if it's a low level. It has to be a significant level above what the oxygen is that we're measuring. Now, that doesn't mean you couldn't measure it by some other method where you could actually extract it and then hit it with an organic chemical that's specific for superoxide versus hydroxyl radical. And I was talking with Jim Davis about that yesterday. There are people that have done that kind of experiment because some organic compounds are more specific for superoxide and hydroxyl radical. So it's conceivable that what's happening is that once the first electron transfer goes to superoxide, you just keep on taking more and more iron out. But those reactive oxygen species could also react with some of the organic compounds and even the organisms themselves. But I suspect their membranes are such that they know how to deal with that. Exactly. So when we're no longer detecting the iron at our detection limit. In this case, the detection limit was about 7 to 5 micromolar in fresh water. We had a really nice oxygen signal. Remember, it's flowing water, too. So it's not staying there. So it's being carried away pretty quickly. And who knows what those reactive oxygen species would do as they go down the hill, right? Other questions? Thank you very much. Thank you. Did you like that? Like some of the pictures? I was hoping you liked the pictures.